All right, this will be an overview of more of the metabolism section of the Unit 4 material. I'm gonna primarily focus on that because the nutrition section is pretty self-explanatory. I will go over just the major nutrients really quickly, mainly just to focus on their uses in the body. So the five major nutrients, let me go back to this slide, are carbohydrates, lipids, which are fats, proteins, vitamins, and minerals. And the main use for carbohydrates in the body is for energy. So all carbohydrates, whether they are monosaccharides or the really large polysaccharides, they can all be broken down into glucose. And glucose is the molecule that is used for energy by the body. Glucose is not used directly by the body cells for energy. Instead, it has to first be converted into ATP. And that is the molecule that can directly be used for energy. Okay, after carbohydrates are lipids, and they can also be used as an energy source, but carbohydrates will always be used first if they are available, and then your body would start to use fats for energy. Lipids also have other uses in the body. They can aid in vitamin absorption. They help with protecting your major organs. You have like pads of adipose tissue that surround major organs just really for physical protection. Protein. So these generally are not used for energy unless absolutely necessary. Proteins have a bunch of different uses in the body. They can act as enzymes. They can act as hormones. Proteins are the antibodies that are involved in the immune system. So they have a lot of different functions, and your body is going to try not to use them for energy because they are needed for so many other really important things. And then vitamins and minerals. So one big difference between vitamins and minerals is that vitamins are organic molecules, whereas minerals are not. They are inorganic. And a lot of vitamins need to be included in your diet in order for you to have an adequate amount of them. There's two different categories of vitamins. Some are fat soluble, some are water soluble. So those that are fat soluble are absorbed along with fats in the digestive tract, and those that are water soluble are absorbed along with the water that's in the digestive tract. All right, so that's just a quick overview of the nutrients. Now we're going to get into the metabolism section, just starting with the main types of reactions that occur in the body. So two of the main reaction types are just catabolism and anabolism. And before we started the digestive system, we reviewed these types of reactions. So catabolism involves breaking a large molecule down into smaller molecules or atoms. Anabolism is the opposite. It's taking smaller molecules or atoms and putting them together to build a larger molecule. So what's generally happening in the digestive tract is catabolism, where you're breaking those nutrients that you've just ingested down into their smaller components so that they can then be absorbed. And then once they get absorbed, your body can use them in various ways. Okay, another really important reaction type, or two reaction types, are oxidation and reduction. And these typically go hand in hand. So when one occurs, the other one is occurring as well with two separate molecules. So when a molecule gets reduced it is gaining electrons, hydrogen ions, and energy. When a molecule is getting oxidized, it is losing those same things. So it's losing or giving up electrons, hydrogen ions, and energy. So a lot of times these reactions are coupled. So when one molecule is losing its electrons and hydrogen ions, it's giving them away to another molecule, and that other molecule is getting reduced. So those reactions are involved quite a bit with metabolism. I am going to skip to just the significance of ATP. So ATP is the molecule that all of your body cells use for energy. They use that directly for energy. And ATP can be made basically in two main ways. It can be made through substrate level phosphorylation, and it can also be made through cellular respiration. So the first way, and this is very quick and easy, but it just doesn't make a lot of ATP. This is the first method of making it. It's called substrate level phosphorylation. So one way that you can make ATP is if you have ADP, which is adenosine diphosphate. You can just add a phosphate group onto it and it becomes adenosine triphosphate, ATP. So basically, you can use any molecule that's in the body that can give up a phosphate group or a phosphate unit, and you can take that and just add it to ADP to create ATP. There is one molecule that is most often used for that, and it's called creatine phosphate. So you would simply just take the phosphate group off of the creatine, put it on ADP, and you can make ATP. So that is the quickest and easiest way. And that's the first way. If your body needs energy, that's the first thing it would do to make ATP. But unfortunately, that doesn't produce a lot of it. So if you are performing an activity that lasts longer than a few seconds, you're going to need to use another method of making ATP. So using creatine phosphate to make ATP is represented by the blue line here on this graph. So that will last for a few seconds, and then the amount of ATP that you're able to make in that way gets totally depleted after a minute. No longer, it doesn't last any longer than that. So if you are continuing to perform any kind of activity, you're going to have to use cellular respiration. So that is what I'm going to talk about next. So cellular respiration involves the breakdown of glucose. So if you use up all of the ATP that you have from using creatine phosphate, then you will start to use glucose, and you will break down glucose to make a lot of ATP. This isn't as quick of a process as substrate level phosphorylation, but it does end up producing a lot more ATP. Cellular respiration involves 4 steps. It involves glycolysis. Then there is a little transition step. Then there is the Krebs cycle, and then the electron transport chain. So a quick overview of glycolysis. This is the very first step, is that this step is actually the only one that doesn't require oxygen to be present. So any process that can occur without oxygen is known as anaerobic. So this is an anaerobic process. It occurs in just the liquid portion of the cytoplasm of cells. That's called the cytosol. So it occurs there. And During this process, you are starting with just one single glucose molecule, which is represented right here. And it is broken down in a series of steps. And ultimately, it results in the production of 2 molecules of pyruvate, which is also known as pyruvic acid. During this process, to get it started, this says phase 1 and energy investment, because you actually have to use up 2 molecules of ATP to get this process going. So initially, you are negative 2 molecules of ATP because you have to use up that energy to get started. But ultimately, glycolysis will produce 4 molecules of ATP. So if it produces 4, but it used up 2 in the beginning, the net gain of ATP would be 2. So that's just 4 minus the 2 that were used up in the beginning. So right away, right after this step, you will have 2 molecules of ATP that can be used right away. But more importantly, what happens is that a couple of coenzymes get reduced during this step. And remember, we talked about oxidation and reduction just a minute ago. When a molecule, in this case, it's a coenzyme, gets reduced, it is given electrons, hydrogen ions, but also energy. So basically, energy has been taken away from the glucose molecule and been given to the coenzymes to store for use later. So when these coenzymes get reduced, the most important thing is that they're given energy, and they hold on to that energy until the last step of cellular respiration. So now what you have are resulting from glycolysis are 2 molecules of pyruvic acid. And those are going to enter the next step of cellular respiration, as long as oxygen is present. Remember that glycolysis is the only step in cellular respiration that can occur without oxygen. So if oxygen is not present, then the process can't continue. And pyruvic acid actually is converted to lactic acid. But if there is oxygen present, and usually there is, then pyruvic acid can continue on to the next step, which is called the oxidation of pyruvic acid. So this step, as well as the next 2 steps of cellular respiration, these occur within the mitochondria. You might remember from A&P1, mitochondria are organelles that are found within your cells. This is what they look like. They have an outer membrane, which is shown in yellow, and they have an inner membrane, which is shown in blue. And that inner membrane is folded, and those folds are called crista. There is a jelly-like substance that's found inside of that inner membrane, and that's called the matrix. So the oxidation of pyruvic acid, this is the second step of cellular respiration, that occurs within the matrix of the mitochondria. So it's in that middle section here where you have that sort of jelly-like stuff. So this step is pretty easy. There's only a couple things that are happening here. You're taking the molecules of pyruvic acid that you have now, and you are just performing a couple of reactions on them. And one of them is oxidizing. So first you have decarboxylation, which is just removing carbon dioxide. But then you have the main step, which is oxidation of pyruvic acid. At the same time, remember, oxidation and reduction are often coupled together. So when you're oxidizing one molecule, you're reducing another. In this case, when you're oxidizing the pyruvic acid, you are reducing this coenzyme right here, NAD. So it becomes NADH plus H. And remember, when you reduce a molecule, you're giving it electrons, hydrogen ions, and energy. So now you've just given this coenzyme energy to store for later. Remember that this also happened during glycolysis. You reduced several coenzymes and you gave them energy to store for later. So that's happening again. And finally, the last step is just adding coenzyme A. So you're just sticking that right on to this molecule here, this 2-carbon molecule. And now this whole thing is called acetyl coenzyme A. So that's the result of the second step. And this step is going to happen to both of the pyruvic acid molecules that you produce during glycolysis. So this shows you one of them, but you have two, remember. So you're going to end up producing 2 molecules of acetyl coenzyme A. During this step, there's no ATP that gets produced at all. So the only thing that's happening with regard to energy is that you are taking some energy away from the pyruvic acid and giving it to this coenzyme by reducing it. So it's going to store that energy for later. All right, the third step is the Krebs cycle. And during this step, you have the acetyl coenzyme A molecules that you just produced. There's 2 of them, remember. So this is showing you one of them. Here is the acetyl coenzyme A right here. So what happens is, actually, first of all, I should say that this is still occurring in the matrix of the mitochondria. So within the matrix, you already have oxaloacetic acid. That is an acid that is already present within the mitochondria. So the acetyl coenzyme A is coming in to join the oxaloacetic acid, and the acetyl group, just that 2-carbon molecule, is being added to this acid. The coenzyme A is just a carrier molecule, so its only job is to drop off this acetyl group with this acid right here, and then it goes away. Nothing else happens to it. So it drops off the acetyl group. The acetyl group combines with oxaloacetic acid, and that produces citric acid. So that is the first acid that gets produced in the Krebs cycle. And that's why this cycle is also sometimes known as the citric acid cycle. With regard to your test, these are the only 2 acids that I want you to know and that I would ask about. So this is the one that's already present in the mitochondria, and this is the first acid that gets produced after the addition of the acetyl group. So from there, the acid, you have citric acid, and it goes through a series of reactions. It just keeps getting modified. It has different names every time a different reaction occurs to it. But ultimately, you can see what happens. If you follow the circle around, the last acid that is produced is oxaloacetic acid. So you're right back to where you started. And that's why oxaloacetic acid is always present in the mitochondria, because it's always the end result of the Krebs cycle. When you go around the cycle, you end up with oxaloacetic acid again. So then you have more of that there waiting for another acetyl coenzyme A molecule to come in. And that whole cycle is going to happen again. And then once again, you'll produce oxaloacetic acid at the end. Okay, a couple of significant things regarding energy that happen in the Krebs cycle. It does produce ATP. So every time you go through the Krebs cycle, there's 1 molecule of ATP that gets produced. Also during the Krebs cycle, you are reducing several coenzymes. The coenzymes are shown in purple. Just like before, we saw NAD before here, and we had NAD in glycolysis as well, getting reduced. So we have 3 of these that get reduced. And remember, that means that they're being given electrons, hydrogen ions, and energy. There's also a second coenzyme involved here. It's FAD. So that one's also getting reduced. Same thing's happening. It's being given energy. And these coenzymes are going to store that energy, and they're going to use it at a later time. So keep in mind that in this step, you had 2 molecules of pyruvic acid going through this step. So you ended up producing 2 molecules of acetyl coenzyme A. So that means there are 2 molecules of acetyl coenzyme A that are going to go through the Krebs cycle. So this is going to happen twice, once with each of the acetyl-CoA molecules. Okay, so those are the first 3 steps of cellular respiration. So far you've produced just a couple of molecules of ATP, but you have reduced a lot of coenzymes. So by reducing them, you've given them energy to store for use in the last step, which is where we are right now. So we're here, the electron transport chain. This is the last step of cellular respiration. And it's in this step where you're taking all of the coenzymes that you reduced in the first 3 steps, and you're basically using the energy from them to make a large quantity of ATP. So up until now, you've made 2 molecules of ATP in glycolysis and 2 molecules of ATP in the Krebs cycle. In the electron transport chain, you're going to make a lot more. So all of the coenzymes that have been reduced along the way, so all of the NADH and all of the FADH molecules are now going to move, if they weren't already, they're going to move into the mitochondria. And the electron transport chain actually takes place along that inner folded membrane of the mitochondria. So I'll go back to this picture for a second. So remember, you have the outer yellow membrane and you have the inner membrane, which is blue. And along the inner membrane, you have, it's like folded. So you have all of these folds here, which are called crista. So let me go back to this picture. This is showing you the inner membrane. So this is that blue membrane that's folded. This is the space that's outside of that blue membrane. And this is the space inside of it where the matrix is. And basically all along that inner folded membrane, there are electron carriers, which is what you see here and here. So these kind of brownish, orangish looking things are electron carrier molecules that are just kind of embedded in that inner membrane. And you can also see the coenzymes that are here within the matrix. These coenzymes give up all the things that they were given when they got reduced. So they're giving up their electrons, their hydrogen ions, and their energy. So those things are going to break off. The electrons that break off get passed from one carrier molecule to the next to the next. So they just pass down these electron carrier molecules. But it's these electrons that actually have the energy. So as the electrons get passed along, they are giving off the energy that they have. And that energy is used to pump the hydrogen ions from within the matrix out to this intermembrane space. This is the space in between the inner and the outer mitochondrial membranes. So now all of the hydrogen ions get pumped out into this space. Once all of the hydrogen ions, let me see. So once all the hydrogen ions are pumped out into that space, there's one final thing that happens. There is only one way to get back in. And all of these hydrogen ions do want to get back into the matrix because anything really wants to spread out. Any atom, any molecule wants to spread out and until equilibrium is reached. So if you have all of the hydrogen ions out in that intermembrane space and now there's none in the matrix, they are naturally going to want to diffuse to sort of spread out, and they're going to diffuse back into the matrix. But the only way back in is through this molecule here, which is also embedded in the inner membrane, and it's called ATP synthase. So these hydrogen ions are slowly moving back in through ATP synthase. And as they move back in, they supply energy, which generates ATP. And I'm going to see if I can show the slideshow because this is an animated slide. So these are the electrons that are being passed from one electron carrier molecule to the next. That's what these blue ovals are. Those are the electron carriers. And as the electrons are being passed, remember I said that they are providing the energy that's used to pump the hydrogen ions from in here out to here, to this space. That's why you saw the hydrogen ions moving out here. And then there's only one way for the hydrogen ions to get back in, and that is through ATP synthase. And as they move back in, they generate ATP. Okay, so I'll show you again. Here's one of the coenzymes that got reduced, NADH. So it's given up its electrons and its hydrogen ions, and now those electrons are passed down. The energy from them is used to pump the hydrogen ions out here. And then as the hydrogen ions are allowed to move back in, they create ATP. Okay. The other coenzyme is FADH2. This is the other coenzyme that was reduced in the first 3 steps of cellular respiration. So it also gives up its electrons and its hydrogen ions. These electrons are again passed down. And as they give up their energy, that allows these hydrogen ions to be pumped out into this space. And then when the hydrogen ions move back in, that allows for the production of ATP. So if you go back to the other slide, each molecule of NADH provides enough energy to generate about 2.5 ATP molecules. And the energy stored within the FADH provides enough energy to produce about 1.5 ATP molecules. So NADH stores a little bit more energy than FADH, and that's why it's able to provide energy or enough energy to produce even more ATP. So one more thing to mention here and then I'm gonna stop the recording, is the significance of having oxygen present within the mitochondria. You definitely have to have oxygen present for this process to occur. And the reason is this. Remember, I said that once you've pumped all of the hydrogen ions out here, they will naturally diffuse from where there's more to where there's less hydrogen ions, because the natural movement of ions and atoms and molecules is to spread out until equilibrium is reached. So if you have way more out here than you have in here within the matrix, then the hydrogen ions from out here are definitely going to move in until equilibrium is reached, and then the movement will stop. So at this point right here in the picture, you can see that we have reached equilibrium. You have an equal number out here and in the matrix. So if the number is equal, then there won't be any movement of hydrogen ions anymore. They're not going to move into the matrix. And that's not good, because you want them to move back in, because every time they move back in, they have to go through ATP synthase, and that generates more ATP. And the whole goal of this is to make as much ATP as possible. So to solve that problem, we have oxygen present in the mitochondria. If oxygen is present, you could see what just happened. It's going to combine with the hydrogen ions as they move in to form water. And then that allows more hydrogen ions to move back in. So let me show you that one more time. Here we're at equilibrium. So hydrogen ions are not going to be able to move through. So these would be stuck out here. And the energy that they have is not going to be used to make more ATP. But because we have oxygen, it can remove some of those hydrogen ions from the matrix to form water. And now that's going to allow the rest of these hydrogen ions to move in. Okay, what would happen next is that another oxygen molecule would bind to these hydrogen ions. They would form water, they would go away, and then these last two would be able to come in. Okay.